All right, well, I'd like to welcome everyone. Thank you for joining us for our first ever virtually on the road program. Although we'd rather be down in Lodi in person, given the current circumstances, this is the safest way to still keep you all informed and for us to also hear from you at the same time. A couple of little rules about Zoom. Remember to keep yourselves muted during the presentations. You're welcome to ask questions by raising the little blue hand, by unmuting yourself and asking a question, or by writing your question in the chat, and then myself or someone else here can ask one of the questions. So either way, any way you guys want to ask a question, please feel free to ask questions. And I think with that, we'll get started. So the first thing we're going to do today is the department chair, David Block, is going to give a quick department update just to let you guys know what's new and sort of give us a little introduction into our first three speakers today. So take it away.
Great. Thanks, Karen. And thanks everybody for attending today's virtual on the road. I'm going to share my screen and show you a few slides to give you an update on the department. All right, can you see that? Okay, good. So what I'd like to do is bring everybody up to date. I know some of you know a lot about the department, but I want to bring you up to date on the latest things that are happening. And of course, a lot of things have been happening in the department, some that we wanted to happen, some that we didn't want to happen, but they're happening anyway. And so I'll kind of bring you up to date. So there are three parts of our mission at UC Davis in Viticulture and Enology, and those three parts are research, teaching, and extension. So I figure what I do is just bring you kind of up to date with what's happening in each of those areas. So for research, I'd say the biggest changes have been that over half of our faculty, 8 of the people you see here, the 15 people you see here, have been with the department for 9 years or fewer. And so we've had a lot of new great faculty members join us in different fields, and you're going to hear from several of them now. In fact, I think everybody on the agenda today, except for me, has been here for 9 years or fewer. And so it's great to have them and all their new ideas and input. And because we have all the disciplines within our department, it does give us the ability to solve complex problems, the kinds of problems that you as people in the industry need us to solve, and that's been great. And of course, these are the people in the Department of Viticulture and Enology, but we also collaborate outside the department throughout UC Davis, throughout California and the country and the world, to bring you the best and the most creative solutions to the problems that you're facing. Those problems include acute problems, things that come up and they need solutions very quickly, things like smoke taint, and smoke affected grapes and wines that we've really only been working on for a couple of years since fires have started affecting the grape and wine industry dramatically in California, through problems that are longer term problems like pests and diseases, which need a solution that's sustainable in terms of cost and the environment. Problems like water. How do we deliver water more efficiently? Because water is always a problem in California agriculture. Do we change the way we sense water status? Do we change the grapevines? Do we find vines that are drought and salt tolerant? And problems like labor. Labor is becoming increasingly an important issue, maybe the most important issue for the wine industry and grape industries in California. And so the idea of how do we do the same thing, the same or better, using fewer people. The idea of mechanization. So all those are ideas that we're working on. And you're going to hear today from several of our faculty members, including 3 of our newest faculty members, on the latest research that they're working on to solve problems that are important to you. For teaching, Our bachelor's program, master's programs have stayed about the same size. We graduate about 40 undergraduates a year with bachelor's degrees in viticulture and enology and about 15 masters in viticulture and enology each year. That's stayed relatively constant for us. The curriculum for both is a core of basic science. It's the bedrock of the cornerstone of our program is having students who understand the fundamentals of what they're doing and can use that knowledge to be creative problem solvers when they get out into industry. All of our program relies on extensive hands-on experience, whether it's in the vineyard or the winery. And so whether it's in a wine chemistry lab or out in the vineyard each of the quarters, our program is using hands-on training in the vineyard and the winery to make sure that our students are able to hit the ground running when they get into industry. So of course the pictures I showed you are the way we like to teach. I will note that last quarter we were completely online because of the COVID pandemic, and likely will be almost or maybe completely online for the fall as well. And we're hoping that that changes. We're trying to change what we do to make sure that any students who missed the hands-on experience in the fall can get it later in their time at Davis. So we're doing our best to do that. Certainly a challenge, as I know it's been a challenge for all of you to deal with. And we're dealing with that as well at the university. Some of the changes that we're putting into the curriculum, aside from COVID, are really important changes that came from focus group meetings with people in industry. In fact, one of our meetings was down in the Lodi area with people that may be on the call on the order of 4 to 5 years ago. And so over the last couple of years, we've tried to implement a lot of those suggestions. And just quickly, things like in our wine production class, Ven 124, we're focusing more on students making their own wines, their own red and white wines, to give them more experience with that. We're in the process of changing the focus of one of our courses on grape production to include more ideas of precision viticulture, and you'll hear from Mason Earles in a little bit. He's the one who's going to be taking that over. That's his area. We've added some new courses, a course on soils and climate, because we heard that was important to people in the industry. Their students had more of a background in that. So we added that. We've taught that 3 times now. We added a course, Ven 127L, on post-fermentation processing. So it's another winery-based class that I've been teaching with our winemaker and winemaking staff to cover everything from blending through packaging. So we've always had a winery course in the fall, and now this is in the spring, so we cover the second half of winemaking. Students can see that all the way through. As of last year, we started a new course, and this is with me and with Karen, who you just heard from. That's a regulatory seminar course, so we bring in people from ABC, TTB, OSHA-related issues, land use, distribution, direct-to-consumer, and so forth, food safety issues, FSMA, and things like that. And again, that was something that we heard that you wanted our students to learn more about. So we've added that course as of last year. And we've started with the Graduate School of Management a business wine-focused business unit that students can take. Alumni can also take that. So if you're interested, you can let us know and give you more information. It's a 1-quarter class. It's mostly online with two days in person during the quarter. We've heard good feedback on it. So if it's something you're interested in, you can let us know, let Karen know or me know, and we can get you more information on that. And we're in the process, this is with Mason and Beth, who you're going to hear from in a bit, of starting a new freshman course where it's very much going through what are the careers that are available to students graduating with a viticulture and enology degree. We'll get our students engaged from the first year they're here. Normally we don't see them as much while they're taking their basic science classes until they get into our viticulture and enology classes in their second and third year. This will allow us to focus more on them in their first year and get them thinking about what they want to do with their degree as they go through. And the third area that we do, third part of our mission is to do extension. And I think many of you know Matt, Anita, and Con are specialists in extension, cooperative extension, and Karen Block, who just announced the program. We now have a new member of our team as of, I guess 2 weeks ago.
July 1st.
Yeah, July 1st. And I think she's on the call, Caroline Furman, who will be helping us with all kinds of logistics for our programs and also communications with you. So you'll start seeing emails from her almost immediately, I would think. This is a program that we couldn't do without a lot of sponsors. And these are, you see the sponsors here who committed to 5 years of support for this extended program. We're doing, in a normal year, 8 to 10 programs on campus, another 4 to 5 on the road events like this one. Grape Days with Matt and with Con at Oakville and Parlier at Kearney. Doing more with social media, especially on Facebook and Twitter. And also bringing people from industry to campus to talk to our students about jobs and careers, opportunities, research projects, and things like that. So that was, if we go back 10 or 12 years ago, we we're probably doing about 1 program a year. And that increased support through the partnership program has helped us to increase that by an order of magnitude. And of course, in COVID, we can't do the kinds of things that we want to do in person, do them safely. And so we've adapted to that as quickly as we could. We just finished 10 weeks in a row of doing a Zoom cast that we call Office Hours with Dave and Anita. Hopefully some of you, I know some of you have been sitting on those, which is great. We're taking a few week break on that while we do some on the road events, virtual on the road events, but hopefully eventually we'll go back to those. And if you have ideas for programs you want to hear through office hours, just let me or Karen know, or any of the Anita, any of us know, so we can start planning those. We have 3 virtual UC Davis on the road events, this one and 2 others planned, and another one in the works for later in the year. And we'll keep doing that as long as we need to, and hopefully as soon as we can get back safely to doing in-person events, we'll do those. I wanted to alert you to this Aggie video, and here's the website here. This is where we're putting the recordings of all our programs, like this one, like all the office hours programs, all 10 of them. And we also have our graduate students or master students, as part of their program, do a review on a certain topic. So here you can see one that's on wine aging and oak. All of these are online as well. So if you go to this website, look for V&E, and there's a channel that you see pictured here, UC Davis Viticulture and Enology Extension. All of these programs are there. Feel free to go look them up, listen to them, and you can listen to them as many times as you want. We'll make sure we put the full link in the chat as we're going through today, so you have that, and it's easy to find. So with that, I just want to remind you, we also have wonderful facilities that we're all looking forward to getting back to. As we do, we invite you to come visit us whenever you can. We're always glad to spend time with you and show you what we're doing in person. We love doing that. And so as we get back, hopefully not in the not too distant future, we hope you'll do that. And so with that, I'd like to thank you for your attention to this update. And there's my email. If you have any comments, you can always send me email. I'm always happy to respond to email and take your comments and questions. So with that, I'm going to stop sharing and I'll turn it back over to Karen.
All right, our first speaker is Megan Bartlett. And Megan, I don't know if you want to start sharing your screen.
Sure.
While I just do the introduction. She's going to be talking about evaluating traits to improve rootstock drought tolerance. She's an assistant professor in the Department of Viticulture and Enology, and we're very grateful to have her speaking today.
Thanks so much, Karen. Give me 1 second, and I should have this in full screen. Let's see how that goes. Can everybody see that all right? Okay, great. So, all right, thanks so much for having me today to come speak, and thank you so much to Karen for organizing. Today I'm going to be talking about some work I've been doing with Andrew Walker and Andrew McElroan in the department to look at traits that we can use to improve rootstock drought tolerance. So improving the drought tolerance of grapevine rootstocks is a really important priority for the industry because California is an ecosystem that naturally goes through cycles of drought. We had one of those recently, and it's likely that that's going to continue to be a problem in the future in California. And also in the future, agriculture is expecting to be competing more with large cities for water resources. So it's more and more likely that we're going to have to keep up our competitiveness as a global wine producer while dealing with less water and trying to stretch the water resources that we have access to as much as possible. And so improving the ability of our grapevines to perform under water stress is going to be really crucial to keep up that productivity, and rootstocks are a really important tool for being able to do that. And so one of the ways that we can actually make rootstocks more drought tolerant is to identify the traits that are really important to rootstock performance under water stress. And so when we know what those traits are that are really critical to rootstock drought tolerance, then those become traits that we can target in breeding efforts and the development of new rootstocks or improving the rootstock varieties that we already have. And so the goal of this project was to really zoom in on some traits that previous work, including in other crop species and in grapevines, had really identified as being very promising candidate traits for improving rootstock drought tolerance. And so what we wanted to see in this experiment was, do these traits actually improve the ability of grapevines to maintain their photosynthesis and their water transport and their growth under water stressed conditions? So in this experiment, what we really did was zoom into some very fine scale cellular traits. And so I want to talk for a second about why we chose that approach. So as an example, Andy Walker in the department has been extremely successful in producing more disease-resistant grape varieties among all of his work with improving rootstocks and improving grape cultivars. And one of the reasons that he's been so successful at generating these disease-resistant cultivars is that in his work, he's actually identified genes that are involved in this disease resistant. So he's able to go through and screen enormous amounts of material for these genes to be able to tell whether this is something promising to keep working on and whether these are varieties that are going to have the disease-resistant traits that we want. But we've really been unable to do that in traits that are related to water use. And that's really slowed down progress in breeding more drought-resistant rootstocks and scions. And so one of the reasons that being able to use this approach of identifying genetic markers and screening through a lot of material is that the genetics underlying drought tolerance is often very complex. If we look at a lot of drought tolerance traits in a plant, we can see that they typically involve aspects of anatomy and structure and molecular signaling that are very complicated and involving organs throughout the plant. And so this makes it really difficult to identify a relatively small number of genes that we can target in breeding efforts. A lot of the traits that we've identified as being important to drought tolerance potentially involve dozens or even hundreds of genes. So it's very difficult to interpret variation when you go out and screen for these genes in new genotypes that you've developed or in germplasm that we have. So what we did in this project is we wanted to really zoom in to some very fine scale traits within the root, with the idea that kind of the more detailed we get, the more focused we get, the less likely there's this enormous complexity of genetic mechanisms involved. So we wanted to zoom in on some very fine scale traits with the hopes that these would have kind of a simpler genetic basis. And so that's going to be the subject of work in the future. But I just wanted to give you this context for why we're really zooming into a very cellular scale in thinking about rootstock drought tolerance. So the traits that we chose are root cellular traits that we expect to impact the sensitivity of the entire rootstock to drought. And so the roots are a major bottleneck for plant water transport, and they become especially important under water stress. So under normal conditions, well-watered conditions, up to 90% of the hydraulic resistance or the resistance to water movement through a grapevine can be coming from the root system below ground. And as the plant becomes more water stressed, that your ability to pull water up the roots becomes even more limiting. So it can account to almost up to 100% of the resistance to water movement through the whole plant is coming from the roots. And it's specifically coming from very small areas of the roots. It's specifically coming from, if you cut a section through the root, It's coming from these living cells that are in between the soil and then the water transport tissue, the xylem. And so those roots are living cells in the root, and they occupy a very narrow range, but they're really where most of the resistance to water movement is coming from in the root. And so if you see this picture kind of in the bottom of the screen, that really narrow layer of soils between the soil and the xylem tissue that brings water up to the rest of the plant is really the most important bottleneck on the ability of the root to transport water under drought. And so what's actually happening that makes these living cells so limiting? And so what's actually happening is, this is work that's been done in the McElroan lab in the department, is that you can see that when the roots start to be water stressed, the root cells actually start to shrink as they lose water, and they actually collapse as water stress becomes more severe. So you can see what this root shrinking looks like in this image in the center of the slide. You can see this is a well-watered root on the left. It's intact. It's really, it's in touch with the soil very well. And then if you look at what that root looks like under a more negative water potential, this is a water potential of 1.5 megapascals, a lot of those cells have completely collapsed and the root is really shriveled up. And so kind of two things are happening. So the root shriveling up is disconnecting it from the soil. So it's really hard for water to move from the soil into the root. And then when you look at what's happening inside the root, that cell collapse and that cell death is actually limiting the ability of water to move through those living cells inside the root. So the picture on the top to the right is showing that pathway of all these cells lined up correctly under well-watered conditions, where water can travel a lot of different pathways through these cells. So it's very easy for water to move from the soil to the water transport tissue that takes it upward to the stem. But then if you look at the bottom picture, this is what starts happening under water stress, where these cells start to collapse and they start to die, and you start losing these pathways for water movement. So it becomes harder and harder to move water, both from the soil into the root, because you're shriveling away from the soil, and inside the root, because you're killing off these pathways that would move water from one part of the root to another. And so we were really interested in this really narrow patch of cells is having such a huge impact on your ability to take up water under drought stress. And so we were interested in what kind of sets this threshold for how stressed you are when this damage starts to happen. So we started to see this happen fairly early under water stress in roots, around a water potential of about negative 0.6 megapascals. So in this experiment, we were interested in, does this threshold for how stressed you are when you start to see this collapse vary across different rootstocks? And do we actually get more drought-tolerant rootstocks if we can change this threshold for when this collapse and this death starts happening? And so how did we actually measure the susceptibility to this level of stress? Oh, sorry, another point I wanted to make is we were talking about specifically the water transport tissue, but we can also think about this through the whole root, where this zone very close to the tip of the root is where a lot of the water uptake is happening. But you also have these cells take on this really important function kind of further up the root in the older root tissue, where they're really important for holding on to stored carbon. So they form starch crystals in these cells. And so that's where a lot of the carbon that the root is using to grow is actually made available to the root. But when these cells start to collapse and start to die under water stress, we don't really know what happens to this starch. Does it actually get used by the root, or is it kind of no longer available for the root? So one of the ideas could be that we are interfering under water stress, this cell death is interfering with water movement and water uptake, or it could actually just be interfering with the amount of resources available to the root in terms of carbon. So what we wanted to look at in this study is, would making these root cells more resistant to collapse under water stress actually improve the drought tolerance of the entire grapevine? Because it would kind of keep up all of these pathways for water movement, and then it also kind of hold on to this storage capacity for carbon. So how did we actually measure the resistance of the root to collapse? And so we can kind of think about this in two different ways. So you can think about, as the root cells are losing water, so kind of the simplest thing that they're doing is that they're actually physically shrinking. So the volume of the root is becoming smaller. And so you can see this is a cross section of the connectivity between the root, is this inner circle, and the soil is the outer circle. So you can see you're actually shrinking away from the soil and you're losing this connectivity with the soil. And so you can measure how the volume of the root is changing as the root becomes more water stressed. But another process that can be really important is the actual water pressure inside of the root cells. And so we can measure that as turgor pressure. And so you can think about this where turgor pressure is the force of water molecules hitting the inside of the cell walls. So you can think about this if you're filling up one of those like plastic kids pools. Once you have enough water in the pool, the sides will actually stand up by itself. And so that's when you consider that the turgor pressure of the water inside that pool is sufficient to hold up the sides of the pool. And so exactly the same process is actually happening inside root cells. When they have sufficient water, the force of that water hitting the inside of the cell wall is actually what's holding up the cells. But then under water stress, when the roots are losing water, they have less and less water actually contributing to this pressure. And so they experience a loss of turgor, and that will actually cause the root cells to collapse when they've lost enough water. So you think about that, you're draining your kid's pool. At some point, you've taken so much water out of the pool, the sides actually fall in. And so that's the same process that's happening in the root cells. And so we wanted to distinguish between these two different processes because there's different ways to build roots that really hold on to volume versus root cells that really hold on to turgor. So we wanted to get an idea of what's happening to both volume and both turgor under water stress in grapevines to give us a greater idea of what specific characteristics we want the root cells to have to kind of lean on either one of these processes. And so we measured the sensitivity of the root volume to water stress through this trait called capacitance. And basically what this measures is you dry out a root and you measure how much the volume of the root changes as water potential becomes more negative. And so a really large capacitance means that your root shrinks really fast as your water potential gets more negative or as your root dries out. So a larger capacitance means you're more susceptible to this shrinking. And then the other thing that we measured was a turgor loss point. And so that's measuring the water potential at which you've lost all this turgor and your root cells have collapsed. And so because turgor is measuring a water potential, a more negative water potential means that you are able to hold on to this turgor under drier conditions. So kind of what we thought would be the best set of traits for improving your rootstock drought tolerance would be a really low capacitance. You do very little shrinkage under water stress, and you would also have a very negative turgor loss point. So you're holding onto your turgor until you reach very stressed conditions. And so what we wanted to do in this study is the first thing we wanted to do was get an idea of whether or not important rootstocks in California actually vary in the sensitivity of their root volume and their root turgor to water stress. We wanted to get an idea, do rootstocks actually vary in their capacitance and turgor loss point? So the idea is do we actually have any variation to work with if we were going to try to breed rootstocks that have improved capacitance or turgor loss points? The other thing we wanted to look at is that roots actually have the ability to really change a lot of aspects of the root under water stress, where they can actually improve their drought tolerance by kind of changing aspects of their cells. So we wanted to get an idea of, are these traits that rootstocks can actually change under water stress to improve their drought tolerance? And then we wanted to look at, does our expectation that a really negative turgor loss point and a really low capacitance or really low shrinkage would actually help grapevines kind of keep up their function during drought? Would they keep taking up water, keep photosynthesizing, and keep growing despite the fact that they're under drought stress? And then finally, so we looked at the first three questions in a pot experiment, because we really wanted to tightly control the amount of water that was available to each of the rootstocks. But we wanted to get an idea of whether these traits are actually capturing your drought tolerance in field conditions. So we looked at rootstocks that are considered to have really different drought performance in field conditions based on field trials. We wanted to ask, are these traits actually predicting whether you're a low, medium, or a high drought tolerant rootstock in field conditions? And so that was the goal of this study. And so what we did is we used one scion variety, which in this case was Chardonnay, and we grafted it onto 8 different rootstocks varieties that are expected to really vary in their drought tolerance. So we use things like 110R, 140 rupestris, Ramsey, and 1103 Paulson as the really drought tolerant rootstocks. These are things that really enhance vigor in the scion under dry conditions. And then we used 420A, 5C, Riparia, and 10114 as kind of low tolerance rootstocks, where these are things where that the scion is really going to have much less vigor under dry conditions if it's grafted onto these. We grew the plants as potted vines in a greenhouse because we wanted to make sure that every variety was receiving exactly the same amount of water. In actual conditions, these varieties can vary a lot in their rooting depth, so you can have the same irrigation coming into the plants, but for example, something like 110R can be accessing a lot more water than something like 10114, just because 110R has a deeper rooting depth. So we did a pot experiment to really control that. And so we weighed and watered these pots 3 times a week to keep everything at the same soil water content. We kept everything, we tried to keep things close to 100% soil water content in the well-watered conditions. We let things dry down to 40 in the drought treatment. And so half the plants were subjected to this well-watered treatment and half were subjected to a drought treatment. And so for each rootstock and irrigation treatment combination, we had 10 plants. And then over the course of the experiment, we let these plants grow for 4 months, and we would destructively harvest some of these plants to measure the root shrinkage and the turgor loss point on the roots. And then we would measure the vines that are left over for their gas exchange. So we'd measure stomatal conductance to get an idea of how open the stomata are. We'd measure photosynthesis. We would get an idea of how stressed the plants are with water potentials. And we would measure the total evapotranspiration of the whole plant just by measuring how much water it lost over the different days. And then at the end of the experiment, we got an idea of how much they grow by harvesting the plants for biomass growth. So this is an example of what these measurements look like, where this is measuring the stomatal conductance for the well-watered plants are in blue, the droughted plants are in black for 10114 over the course of the experiment. And then this is what water potential looked like. And so what did we actually find? So first we found that these traits do actually vary a lot among rootstocks. So I'm going to focus on that finding first. Where here the well-watered values are shown as blue bars and the droughted values are shown as gray bars. And so this is showing the variation in these 2 traits among all of the rootstocks under well-watered and water-stressed conditions. So first, just focusing on the well-watered conditions, you can see that the rootstocks varied a lot in how negative this turgor loss point was, where things like 420A and 5C had very drought-sensitive values, so they lost all of their turgor and experienced cell collapse when water potentials were still pretty high, pretty close to 0. And then things like 110R and 1103 Paulson were able to hold on to their turgor until water stress was a lot more severe. And then looking at the amount of root shrinkage, we're also seeing a lot of variation, where again, things like 420A and 5C experienced a lot of shrinkage under a certain decline in water potential. And things like 110R and 1103 Paulson didn't experience nearly as much root shrinkage. So they held on to a lot more of their root volume. And so that's a really great finding that there's actually variation to work with in the rootstocks that we have. So if we wanted to breed for certain traits, we actually have a good amount of genotypic and diversity in material that we have. So the other thing I wanted to look at was, can rootstocks actually adjust these traits to make the roots more drought tolerant under water stress? Do they actually have this capacity with these traits? And we saw that yes, most of the rootstocks do have this capacity to make these traits more drought tolerant under water stress. So they're actually changing aspects of their cell chemistry or their cell structure to make these traits better conditioned to water stress. So you can see that the turgor loss points are becoming more negative in all of these varieties. So they're holding on to turgor until they reach greater water stress, and they're reducing the sensitivity of the root volume to water stress. So they're doing a lot less shrinkage after they've been allowed to kind of get used to this water stress and change their root traits to adjust to that. And so that's also a really great point, which is this is showing us that the roots actually have this capacity to adjust in response to water stress. And it also gives us guidance as to how we would screen for this in the future, where we know that just looking at these traits under well-watered conditions is not going to give us the information that we need to actually figure out how tolerant these traits are under actual drought conditions. So that's informative for figuring out how we would screen for these traits in the future. And then our other finding was that these traits, specifically traits related to the root volume, are actually helpful in maintaining your stomatal conductance and your carbon gain through your photosynthesis during drought. So this is the average of, on the left is stomatal conductance, or how much water is coming out of the stomata, sorry, both of these are stomatal conductance. For each of the rootstocks over the course of the experiment, each point is the average over the whole experiment, and each point represents a different rootstock. The blue points are the well-watered plants, and the black points are the water-stressed plants. The plot on the left is showing the relationship with turgor loss point, and the plot on the right is showing the relationship with this capacitance or this shrinkage. And so what we're seeing is we didn't really find a relationship between the turgor loss point. Holding onto your turgor wasn't such a big deal, but the amount of shrinkage that your roots experienced actually had a really big effect on your gas exchange under drought. Where under water stress, the roots that experienced more shrinkage, that had a higher capacitance, actually had to shut their stomata and reduce the amount of water transport that they did. And so that was really well correlated in the water stress conditions, and in the well-watered conditions, this didn't really seem to matter so much for your stomatal conductance. So looking at photosynthesis, we saw the same relationship, where the more your roots collapsed and lost volume, the less photosynthesis they were able to keep up under water-stressed conditions. And this really had less an effect under well-watered conditions, which we might expect, since they're not going to be experiencing much of a cell collapse. So this showed that actually these rootstock traits are predictive for how well your vine performs under water stress, but there's kind of a catch. So the next thing we looked at was the effects on growth. So one of these issues is that we looked at what's the effect of these traits on your root growth, to the right, and your canopy area to the left. So we actually found in this case that they all had basically the same vigor. This is just showing the relationship to turgor loss point. The capacitance is the same, where the root traits didn't really affect your scion vigor very much. They did affect your root area, where the varieties that had that held on to their turgor more were able to keep up more root growth. But this didn't actually affect your whole plant very much in this experiment. And this is kind of the finding that sort of makes all of these results a little bit more complicated, where we then compared the traits that we measured in these potted plants to how these rootstocks are categorized for their drought tolerance in field conditions. And so that goes back to the ranking I was talking about in the beginning, where things like 110R and 1103 are things that really increase scion vigor under drought stress compared to varieties like 420 and 5C. So those are considered to be very drought tolerant rootstocks in field conditions. And so we expected to see that these water stressed values for these traits would be very important to predicting how drought tolerant you are in field conditions. But actually for turgor loss point, we saw that the only thing that really predicted how well these plants did was the values in well-watered conditions, where we saw that the varieties that are drought tolerant have a more negative turgor loss point in well-watered conditions, but very weirdly, they have a less negative turgor loss point under drought stressed conditions, which is, we would expect that things like 110R and 1103 Paulson would actually be experiencing more root collapse under water stressed conditions, because they're not as good at this adjustment as these other varieties. And it was the same story with capacitance, where the less root shrinkage they did in the well-watered conditions, the more drought tolerant they are in the field. But the capacitance under water stressed conditions actually had no effect on how drought tolerant the rootstock is in field conditions. So this kind of brought up a lot of questions about what's actually different between how these traits are affecting how you perform in a potted plant experiment versus in a field condition. And so this is what I think is happening, where this is an example of what your plant looks like if it's in a pot, where your whole root zone is kind of constrained to have sort of the same amount of water stress. In field conditions, grapevine roots can be very deeply rooted, but when you're in a pot, you're kind of experiencing a more uniform condition. And so what's happening is that these roots that are at the bottom of the pot are experiencing kind of pretty similar water stress conditions at the top of the pot. And so if that plant is good at keeping its volume under water stressed conditions, all of the roots are water stressed. And so that means that the whole root system is better at taking up water. So being able to lower your capacitance under water stressed conditions is really beneficial for you when you're growing in a pot, when your whole rooting zone is water stressed. But what actually happens in a field condition is, because these grapevines are so deeply rooted, most of them, what's happening is that maybe you're water stressed at the top of your soil layer, but then you actually have an abundant amount of water at the bottom of your soil layer. So you have a really complex environment. And so in that case, what we actually found is that the rootstocks that perform best in this condition, when the soil's dry at the top and wet at the bottom, are actually these rootstocks that do less adjustment in dry conditions. So what they're doing is they're maintaining their volume very well in these well-watered conditions where the soil is wet. So they're really keeping up that water transport from these wet soil layers, but they're allowing the rootstocks, the roots to collapse and die in these dry conditions up at the top of the rooting zone. So they're actually allowing these roots to become worse off than the roots that are in this kind of deeper, wetter soil layer at the bottom. And so if you look at the first condition, this is what's happening in our pot experiment where everything is exposed to stress. And so this is actually the best set of traits when we're thinking about roots growing in actual vineyard conditions where there's a really variable environment. But what does this mean? So why is that actually beneficial to let these roots collapse and potentially die off up at the top of the soil layer? That seems very contradictory. It seems like being better connected to the entire root zone would actually improve your whole drought tolerance. And so the reason I think this is happening is that these rootstocks like 110R and 1103 Paulson are sacrificing the roots in the dry areas of the soil in order to improve the function of the roots in the wet soil. And so one of the things that happens when you have roots in dry soil and roots in wet soil is that the roots in the dry soil can actually pull water from the roots in the wet soil. And that increases the lifespan of the roots in the dry soil because they have access to water, they're staying alive for longer, which seems like it should only be a good thing, right? You have more living roots throughout the whole soil zone. But actually the respiration of roots, or the amount of carbon that the roots are using up, is extremely high in a grapevine, where the amount of carbon that the roots are using up can actually account for up to 70% of the photosynthesis of the grapevine. And that especially becomes true when the grapevines are water stressed and they're doing less photosynthesis. So actually keeping these roots alive in dry soil is potentially very expensive. And so what we're seeing is that the benefit of keeping these roots alive in the dry soil, it may actually be less than the costs of having to pay carbon to keep these roots alive. That these really drought tolerant rootstocks are deliberately allowing these roots to become damaged, and they're dumping them off in order to concentrate resources on your root growth in your wet soil. And so there's some results from Andy Walker's group that kind of support this. So this is looking at a cross-section through a pot, and the lines are representing the root growth throughout the pot in a drought stressed, in a variety that's very drought sensitive, Riparia, versus a variety that's very drought tolerant, like 110R. And so what Andy was finding is that the drought tolerant variety really rapidly recovered its water uptake after it was irrigated again, because it really rapidly proliferated roots throughout the pot. It grew a lot more root area, new root area than the drought sensitive rootstock did. And so this mechanism of actually allowing the roots in the dry soil to collapse might actually be a mechanism that helps the drought tolerant rootstocks do this, where they're dumping off these wasteful roots that aren't helping them anymore in order to grow more root material into deeper, wetter soil layers. So that is kind of the basis of what we found right now, which is actually really surprising, because when people talk about breeding more drought tolerant roots and other crop species, they tend to think that more drought tolerant is always better, that being able to do more adjustment is better, having a more negative turtle loss point or less shrinkage is always going to be better. And we should always be shooting for those trait values. But what we were finding is that the context matters a lot. Being able to screen these in vineyard conditions is going to give you a very different answer from screening these in pot conditions. We have to keep this complex environment in mind when we think about what traits actually improve a grapevine drought tolerance. And the other thing we found is that this idea that more is always better doesn't necessarily seem to be true for this root drought tolerance. That actually this strategy of keeping the roots connected to the wet soil and allowing them to get damaged and dumping them in the dry soil was what really made the rootstocks like 110R and 1103 Paulson more drought tolerant. And so when we think about what traits do we want to target if we are developing rootstocks for drier conditions, we need a better understanding of under what conditions is dumping the roots in dry soil a better strategy than keeping these roots functional. Is this always going to be true? For example, we can think about how this relates to soil type. This idea that dumping roots in dry soil to grow more roots from wet soil depends on the idea that we actually have layers of wet soil available for grapevines. Is this true everywhere across viticultural areas? Is this going to continue to be true as we have conditions becoming warmer and drier, and therefore there might be more evaporation directly from the soil? So we need a better idea of under what conditions the soil is actually going to have wet enough layers to make this strategy of dumping roots worth it. We also need to think about irrigation management, where the soil in the upper layer of the soil in the uppermost layers is the soil that dries out the fastest, but that's also the soil where the irrigation directly comes in. So we need to think about, does it actually always benefit the roots to dump the roots in this upper drier soil layer and grow new roots in response to irrigation? Or if we change our irrigation frequency or our irrigation strategies, do we actually get better performance by holding on to those roots and using these more drought tolerant trait values? So how is this strategy of dumping roots going to change and how effective it is as we change how we use irrigation? Kind of the next question is, can we actually improve on these trait values?
Megan?
Could we push to have these roots become even more susceptible to dumping them, or would it help to have a kind of a more intermediate strategy where we hold on to some of these roots and not all of them? And then finally, are there other traits that it would be effective to target in coordination with this root capacitance or this root shrinkage to improve rootstock drought tolerance? Where we were thinking about this really narrow zone of cells between the soil and the water transport tissue to the above ground part of the plant. But could we also think about that tissue that's doing that water transport above ground? And could we think about the roots more holistically and get a better performance than just from focusing on these cells right now? Okay, so thank you so much. I want to acknowledge that this project was supported by the American Vineyard Foundation, and I really appreciate their support for this. I was supported by the department and the college, and also a lot of really excellent collaborators in the Walker Lab and the McElroy Lab. So thank you all so much for attending, and I'm really happy to take any questions.
I think we're going to wait on the questions. Can you all hear me?
Yep.
Okay. So I think we'll wait on the questions, and we'll put those off until after the first three speakers have had a chance to speak. So if Mason, if you would like to share your slides.
Are you seeing it?
Yep.
Cool. Okay, great.
Great. So Mason is going to talk about AI-enabled precision viticulture. He's an assistant professor in the Department of Viticulture and Enology, and also in biological and agricultural engineering. And we welcome him. He just started last fall, and go ahead.
All right, thanks, Karen. Hi, guys. I'm very excited to be able to give you a quick update on what our lab has been up to so far, and hopefully get some of you excited enough to follow up with some ideas on where we could kind of aim the compass going forward. So I'm going to talk a bit about specifically AI-enabled precision viticulture. And as far as my background goes, I have a bit of work both in the computational experimental side of plant physiology, but I've also worked in industry as a data scientist and machine learning engineer. So I'm trying to bring those two together and think about this in the context of viticulture, and then also somewhat more generally in terms of agriculture. So the general idea that we're interested in understanding in the lab is how we can help great producers, you guys, sustainably manage vineyards to maximize yield and quality, and then at the same time minimize resource consumption and waste. And we want to know how AI, and that is artificial intelligence, might be able to help with that on the sensing side all the way through the mechanization and understanding crops more generally. So from a broader point of view, in terms of AI-enabled agriculture, there are a couple big challenges that we face, and viticulture falls within the same set of challenges. First, we have a lot of variability in terms of production conditions. This ranges across crop, cultivar, environment, and management types of conditions. So that is, there's a lot that we have to sense and see through in order to get a signal through the noise. So the second challenge that we're facing is that we often operate in limited internet bandwidth, memory, and power supply. So we're operating in remote areas, is what that basically boils down to. Challenge 3 is that really the technology adoption for these types of systems requires that agricultural producers actually have confidence in the system, right? So that's really important, that you want to use it, that you trust it, and that you believe that when it makes a prediction, it's something you can be confident in, and it's not going to be a complete black box. It's something that can be understood. So as far as a general roadmap, and I'm going to dive into specific projects, but this is kind of the outline for some of the work that's going on both in my lab and across the university in terms of AI-enabled agriculture. And I'm not going to go into great detail on this, but the general idea is that we've got sort of this larger scale set of objectives that researchers across the university, we're all working on together to try to understand how we can bring together things such as 3D crop models through environmental and climatic data with AI-enabled sensors, and bring those into kind of new types of models targeted at agriculture specifically, so that we can predict things that actually matter to growers, such as yield and quality, and also understand how we can feed back into management types of actions, either through kind of collaborations or mechanization processes with robotic types of systems. So diving into a little chunk of this, I want to talk a bit about more concretely a set of projects that we've been working on in the lab for the last 9 months now. This has to do with AI-enabled sensing, and specifically developing models from these data that we're sensing and trying to predict yield, ultimately yield and forecast the time to ripeness. So this is not by no means complete, but we've made a lot of headway so far, and we're excited to keep going forward, hopefully find some collaborators through Lodi to work with on this as well. Some of the work you'll see actually did happen right outside of Lodi. So the specific project that I'm going to talk mostly about here is related to AI-enabled non-destructive yield mapping in vineyards. This is in collaboration with a number of other folks, both at UC Davis, the USDA, and then also Ian J. Gallo. So here are the pictures of the three, 2 grad students and 1 undergrad who have done most of the legwork here, Alex Pranav and Jung Hao. So I'm going to dive in to an area that's probably pretty close to where some of you all are at. So this is Borden Hills. This is a Ian J. Gallo, I believe, owned in this case, vineyard. And we have, please correct me if I'm wrong, we have this location here that we'll zoom into, and you can see there's quite a bit of all of this is vineyard production. This is in collaboration also, as I mentioned, with Beth Forrestel, who you'll hear from next. Zooming in some more, here's where we landed, right in one of these vineyards. And the idea with this particular project is large-scale commercial operations, and anyone who uses a vineyard manager, I guess a medium and large scale, often use these sort of mechanical grape harvesting equipment, right? And so some of these folks, such as Gallo and others, have these harvesters equipped with load cells. So that is specifically, they have the ability to go in and roughly do yield mapping on how much they're producing on a per meter basis. So they can get, as you probably a lot of you have seen, these yield maps that you can potentially use to drive your management types of decisions. One of the challenges with having a yield map from a harvester is that it's produced when you're harvesting. So the actions that you can actually take based on that type of information really happen there are some actions you can take immediately, but most of what you can do is for planning for the next year. And over the period of time where you get more and more of this data, you have a better sense for where there might be high and low production regions in general, and things like that. But what we really are interested in is having a way to forecast yield, right? I've been told if you could know going into veraison what the yield is going to be, that would be particularly beneficial as opposed to the day that you harvest. So that's what we're particularly interested in. And what we did was work with Gallo, and this is a aerial view. Every one of these points is a yield value ranging from, I should have a legend here, but the lower values of yield are in purple-ish blue, all the way up to the higher values, which are yellow. And this is collected from the mechanical harvester and plotted using high resolution GPS about every meter. And what you end up with is tons per acre, right, at every one of these points on a specific kind of per meter space. So you have to calibrate this. This is one of the tricky things about these yield monitors. In the first place is you still have to weigh them and calibrate the bins at the end of the harvesting process to calibrate the yield monitor. You have to do this pretty regularly. So what we did was the first thing we went out and started saying, well, we have a couple weeks left to harvest whenever I started. What can we throw together pretty quickly so that we have some data to work with throughout the winter? So we went out, Alex put together a great system, low cost. This was a few hundred, well, I guess about $300 camera, what's called a little computer, basically a Raspberry Pi, some GPS unit, put all this together. And we drove through a number of rows where they had collected this yield. They would be collecting this harvest data in a few weeks. This is some of the hardware that we used. I'm going to skip through that. And what we end up with is just 6 rows. I guess there are 5 rows here where we have both sides of the row. So we went up and down on a given row, and we have video. And they're spatially located. We drove about 4 miles per hour, and this is what the video looks like. If you can get a sense for this, I'm going to stop it here in case it's choppy. But you can see that basically we have, it's pretty occluded. It's kind of a messy looking canopy. And so as we move through, that's one of the challenges that we face, right? Not only that, whenever we get this video data, we have a lot of images that aren't useful for what we're trying to do. So what we want is this one in the middle. We don't want these 2 on the sides. So the way this works in the artificial intelligence world is we have the most common way of doing this is called supervised learning, which is a human goes in and just draws in the computer and says, this is where the thing is I'm interested in this case, grapes. So Alex went into about 120 images and drew around the grapes and said, this is where they're at. Now, give you a point of reference, just for those 6 videos, we have probably something like 15, 20, 25,000 images, right? So he did about 120 of those 20,000, and that took him about 6 hours total of his time to do. And then we trained a model. And again, I won't go into any details about this, but we trained some of the state-of-the-art models out there in the ability to object detection. So to be able to detect objects in images or videos. We built sort of what we call a pipeline, data pipeline, to be able to plug in this data and output something useful, in this case, where the grapes are at. And I will skip over this, but we got pretty good performance on these models, and I'll give you some intuition for what I mean by that. So there are 2 different models. I wouldn't focus too much. What you can just look at is say, look, where are the grapes at? You can see that it's actually doing a pretty good job of detecting when I see a grape in an image, where the grapes are at. The only difference in these 2 are this one's generating this sort of polygon, this blob around the grapes. This one's just doing boxes. So you say, okay, well, how well is it doing? We held out some of these images that the model had never seen, but that Alex had drawn on top of and plotted these 2 here. And so this is the annotated area versus the predicted area. And there's a pretty good correspondence between what Alex drew on the image and what the model predicted on these images it had never seen before. So we were confident that it had reasonable performance internally, same with the bounding boxes. But what the hard part is going from pixels, so the image, to the kilograms, right? And so that's where we have this yield data, and we say, okay, well, can we actually predict from something out of our model, the actual yield? Not just, here's your grapes, here are your grapes, but something more along the lines of, here's the mass on a per hectare basis or a per square meter basis. And long story short, we do not see a good relationship, right? So that was the first thing we saw. So we said, oh dear, what's going on here? So we went back and said, well, who's wrong, us or the yield monitor? Because we've heard a lot of mixed reviews about yield monitors. So we went back to Brent Sams, who's at Gallo, and said, okay, did you do any hand harvesting? That's our gold standard, right? Can you show us the relationship between your yield monitor and hand harvesting? He said, yeah, I did some. And so this is the plot of that. You can see there's a decent relationship. He did no smoothing. This is completely raw data. And so if he did some smoothing, I think it would get even better to show that the yield monitor and the hand harvest are related, better than our predictions. So we have to conclude, well, it seems like we're the ones who are wrong. And then we start asking why, what's going on, what makes us, or what's going wrong here? So if you go in and start looking at these images, we wanted to do a deeper dive into what might be happening. We saw some discrepancies at the 2 ends where we were always way off at the beginning and the end of the field. And you start looking through these and you start identifying potential sources of error. And so there's things like the distance from the camera. Some grapes are close to the camera, some are further away. We don't have depth in there right now. There's occlusion and sparsity. So occlusion being some grapes cover other grapes, leaves cover grapes, and so on. You also end up with grapes. When you have a sparse canopy, you might have grapes that are visible on the other side of the canopy through the canopy that are being double counted. So these are all potential issues of just using standard RGB, red, green, blue imagery. And then you even have averaging across sides. Should we use one side or the other? The yield monitor is kind of doing both. So how do we account for that? And the real question is how we can overcome these obstacles. And so what we started doing now is working on, we call it auxiliary data inputs and building ag-specific AI models. And so in this case, we're starting to, working with Beth Forrestel and Stavros Vizhukas, put together a second version of the sensing kit. And so part of this is to strap it onto a robot and drive it through RMI's Vineyards, which I'm not gonna show this whole video, but you can just get a sense for how this works. And it's just driving through every week and collecting data throughout the RMI International Vineyard block, where Beth is monitoring at the same time a lot of the phenology, yield, the quality and chemistry parameters that ultimately we would like to be able to sense earlier on in the season. So we get not only that, we're doing this with more sensors. So now we have infrared, we have depth, and this is all in a camera that cost about $200. So what we're trying to do is build this low-cost system that we can get more of this information and we can start addressing some of those challenges that I identified earlier. And the second thing that we're working on now is trying to build on this idea that most of these tech companies do, which is just get a lot more training data. So it's the question is, how do you do that? So the way we're working on this is we want to be able to deploy a low-cost tractor sensing system on existing tractors and integrate into existing operations. So we started talking with different folks within industry and asking, well, what seems to be the best way to integrate into existing operations? Where are you already going out the most? And what are you doing? It turns out it seems that dusting, sulfur dusting, is one that happens a lot pre-veraison, about every 7 to 10 days, has the challenge of happening at night. Maybe that's a blessing or a curse. And the speeds really can range from 4 miles per hour to a number of people I talked to going 8 to 10 miles per hour. So it sounds like you can fly on this tractor through the vineyard and do this. To give you a sense, we're thinking of things about mounting positions. So we're looking at how, where to mount this sensing kit. We've measured a few different potential locations. We're looking at things such as vibrations and motion, trying to understand how you deal with the fact that a tractor vibrates a ton. So getting a good image is not the same as doing it on your Tesla on the highway. So we have to start thinking about that as well. And we have to integrate all of this into a system that can predict this scale of data across many different vineyards. So we want to team up with we're teaming up with both, again, Gallo and other vineyard management companies to get the actual harvester data to train these models. And the idea is if we can train them on this type of data, maybe they can form the basis of other models that could be useful to other folks who may not have the capacity to do yield monitoring on their harvest, but through a simpler low-cost system. And so to do that, in kind of bringing it to a conclusion here, we're looking at different types of models from the AI community that are emerging now that we think might be able to handle many types of data coming in. And being able to add this depth channel to our camera through this low-cost sensing, trying to deal with some of the occlusion and sparsity, again, averaging across size. These are all questions we're working on now. And that, I wanted to just drop this last piece. We're also doing some stuff related to generating data in the first place using synthetic vineyards. So that's something you can actually train AI models using synthetic 3D models like video games, and you can make predictions in real vineyards. And we see actually pretty good predictions when the models had never seen a real vineyard. We just show it video game grapes, and then you go out in the field and you take a picture and you say, where are the grapes? It can find the grapes pretty well with never seeing actual images. So that is about it. This is the roadmap we're kind of working on again, and I am looking forward to hearing from hopefully each and every one of you about what you think is great or not so great about what I've talked about here. So thanks a lot. Here.
Thank you very much. So we'll take questions again after the third of the first 3 talks. So Beth, if you want to share your slides. And I'll sort of start the intro while you're doing that. So we have Beth Forrestel. She's also an assistant professor in the Department of Viticulture and Enology. And the title of her talk today is Strategies for Adapting Viticulture to Heat Waves and Future Warming. Are you unmuted?
Sorry. Okay. Thank you, Ken. Minor details. I seem to have trouble with those lately. Thank you, Karen, and thank you, Dave, for organizing this, and Anita. And I am very excited to be here. And thank you to Stephanie Bolton, too, who's been great in terms of communication with Lodi and the growers out there. So Lodi is actually a site I'm at no longer me, but I was at almost every single week, and we work at regularly. And so it's great that Mason introduced it. And I'll talk about a little bit more about exactly what we're doing out there. So it's exciting to talk to all of you about this. So this is not something that is unknown, but this notion of having these warming day or mean temperatures in terms of increases in mean temperatures over the past several decades. But one thing that is generally less focused on in terms of climate change research is understanding how changes in maximum temperatures and minimum temperatures and extreme climate changes or extreme climate events are changing. And this is something that is pretty critical to viticulture in general. And so,
Beth, we're not seeing the theater full screen view.
are you seeing the slides change?
No.
Okay.
Now it did. And now it went to the right slide, but we're still
now you see the side.
We're seeing the deck on the side, yeah.
Okay. If it's okay, can I just keep going like this? Will it bother anyone to see the deck on the side? Because I don't know why I'm having these issues.
Yeah, that's fine. Just, you'll have to click on the next slide in order for it probably to advance, yeah.
That's fine. I apologize for that.
No worries.
So all I was saying is that there's changes in average warming and that extreme events are on the increase as well, and actually at a more rapid pace than mean temperature change. And that's largely not a focus of climate change research. And so this is really relevant to viticulture. And one thing that I'm going to do is talk about heat waves. And in the context of Lodi, the way I define a heat wave is relative to the local climate. So when you see a 3-day period with temperatures over 100 degrees, just as we did this past weekend, that is what we're defining as a heat wave. And you can see that this is on the left-hand part of this panel. You can see the change in the number of 3-day heat waves across California at the end of the century relative to current times. And it's pretty dramatic. And Lodi right here is actually in an area that will see quite a bit of increase. And then on your right, we have to think about too, when we consider drought and things like what Megan was talking about, is how there's actually, coupled with this extreme heat, is going to be this pressure in terms of evaporative demand. And so this is showing you the increase in potential evapotranspiration, both from the plant and evaporating from the soil. So in Lodi, this is showing you what we'll see. And this is a climate scenario where business as usual will be maintained, which is what we're on track for right now. And you can see this is sort of the current number of heat waves. There aren't that many. And so right now, this is not number of heat waves. This is number of days in the year that's above 38 Celsius or 100 degrees Fahrenheit. So it's not just heat waves, it's just warm, extremely warm days. But you can see that this is pretty dramatically going to increase over the coming decades. And so this is something that, as far as management considerations, we're going to have these non-analogous events that are really critical to be able to deal with. And so work in my lab is, I have 2 lines of research. I should say I come from an evolutionary ecology background, so I really think about physiological and evolutionary basis for adaptation and climate change. But in the context of viticulture, I'm really thinking about sort of short-term adaptive measures. So how can we mitigate the damage of heat waves using irrigation and potentially other strategies like misting or shade? My focus right now is irrigation, characterizing thermal tolerance of different cultivars, and experimental manipulation of this. And then that leads to the long-term adaptive measures. So how can we generate new metrics to actually assess where cultivars are best adapted, and how can we choose cultivars for the future? So I'm just gonna scroll down here. As part of this, one thing that we've started doing is actually assessing what growers are currently doing. And this is in collaboration with the USDA Climate Hub and postdoc Lauren Parker. We are continuing this survey. I'm sure some of you have seen me talk about this before, but we're continuing this survey into the next month. We've gotten really nice responses and I'll show you some of the preliminary results, but actually we don't have great. We could use more in the Lodi area. So it would be great to have you participate. This is the link, and I'll bring it up at the end, and I'll put it in the chat as well. So just to give you an idea of what we're finding, this is showing you in the upper left-hand corner what varieties people are growing. And so this is all grower data. And not surprisingly, a large portion is Cabernet, almost 50%. It's one of the most widely grown, and next is Chardonnay. And this is data from all over California. And then also too, we have a lot of smaller growers that have participated to date. And we actually have a split between very small growers, less than 50 acres, and then about 28% is 500 or more acres. And what I think is really important to note, so this is only a tiny portion of the survey, although I should say it only takes 10 minutes. It's very quick. But when we asked what problems people foresaw as being the most relevant to viticulture and managing their vineyards, the two that, so the color you see blue is not a problem, all the way to purple, which is devastating. They perceive this as devastating. And this is from 43 respondents. That actually drought and warmer daytime temperatures, extreme temperatures, were the two that were most concerning, and this is across California. So these are really serious issues that growers really want to understand how to mitigate. And so the work that I'm doing at Borden Hills and Lodi in collaboration with E&J Gallo at a site with precision irrigation or variable rate drip irrigation at a 30 by 30 meter scale is this block of Cabernet Sauvignon on 1103P. It's mechanized, leaf pruning, leafing and harvest. And I should say that the person who has done the vast majority of this work is a master's student of mine. She actually handed her thesis in, or not handed in, but finished it, and it's beautiful yesterday, which is really, I'm super proud. It's an amazing job, and she still continues to work this summer. And so what we have done, and we started this last year and we're continuing to sample this year and look at long-term effects. And what we've done is these 30 by 30 meter pixels, which have now this year been expanded to 60 by 60 meter pixels, is we have 3 different treatments. We only manipulate irrigation or have these treatments during heat waves. So this is not a manipulation across the season. And so we're keeping it baseline about 60% ET, 2 times that amount, and this is a day prior to the heat wave, through the heat wave, and then 3 times that. And we're looking at physiological, chemical, and response. We actually make wine from this fruit as well. And so this is just to give you an idea of what we saw last year. And so there were 2 heat waves. We've already had 2 this year. So we had one during flowering thus far, and one just now we're at about 5 to 7 bricks out there right now. But last year we had one at the end of August, and that was 3 days. And then we had a more extreme heat wave later in August. So that was post-veraison. So we had one pre-veraison and one post-veraison. That's important as I move forward to remember that. And as you can see, this is the baseline irrigation that is gone on throughout the season. This is the daily applied irrigation in gallons per acre. And then we have our baseline during the heat wave, and then 2 times and 3 times that. And then you can see this is the temperature here on the right side of the axis. And so many growers use water potentials, and this is stem water potential, but leaf water potential largely followed the same trend that we're seeing. And so again, these are the 2 heat waves in July and then August. And what was pretty remarkable and surprising to us is that they're actually the lower, so the more negative water potentials, the more stressed the plants did not get very stressed in response to these heat waves as far as water potentials go. So this is right here around, they even got up to -7 bars, and they were not stressed. And so this is what, just I want to emphasize that. And there weren't huge treatment differences except in the recovery. You saw significant differences. But we did see responses in terms of carbon assimilation. So this is net photosynthesis on the left, and then stomatal conductance here on the right. And so at the bottom here is leaf temperature. that we were measuring. And you can see the green is always going to be the baseline. The green you saw, there was a reduction in stomatoconductance, sorry, over here, and a reduction in net photosynthesis. And you got really quite warm temperatures. You got up to a 48 degrees Celsius, which I'm going to not quickly convert that, but that's 110 or more. It's very warm. And these are sunlit leaves, so it's not the temperature of the berry. And this year, what we're doing is actually using thermocouples to continuously monitor berry temperature and leaf temperature in the canopy, as well as use IRTs above and model. And so we'll be able to actually look at continuously what the berries are experiencing as well. And so we did see these responses, but I should say, and I'm not showing this, but we didn't see any issues with recovery. So by a few days after the heat wave event, they were totally fine. Everything seemed normal in terms of photosynthetic responses, and you couldn't see a treatment difference. And this was only 2 heat waves, so. But what was really interesting is that we actually saw a pretty, a significant response in terms of both yield. So we had both a reduction in yield for the baseline, where we didn't up the amount of water before and during the heat wave. And so we had lower yield in terms of kilograms per vine and grapes per cluster. So, and we had smaller berries and smaller clusters. And there was no difference between the 2X and 3X, a slight decline in yield in the 3X. And this is something to pay attention to as I walk through the chemistry, is that we actually saw a negative effect of having excess amount of water. And this is something that I'd be interested to talk about growers in more detail and some of their thoughts around that. So in terms of bricks, again, we had in the baseline where there was a reduced water, higher temperature, but declined photosynthesis and carbon assimilation. So I was actually a little surprised to see this. It could have slightly been due to dehydration, and it reached physiological maturity earlier too. We saw that bricks were higher generally in the baseline and no difference between the 2X and 3X. So we actually harvested, our target bricks was 25, or we harvested a week earlier in that block. We also saw an increase in pH and a decrease in titratable acidity. And again, we generally found not a significant difference between the 2X and 3X, a slight difference in pH. And then with phenolic, so I'm very interested in understanding how climate and weather actually affects secondary chemistry. So we often, especially in ecology in terms of modeling responsive grapevines to climate change, we think about either harvest dates or bricks. But really, we care a lot about phenolics for making the wine. So what was really interesting is that we had a pretty significant effect on total anthocyanins. So again, we have the baseline being the one that suffered the most in terms of anthocyanins, and then the 2X, which had the highest concentration of anthocyanins. And similarly with tannins, we had the greatest amount of tannins in the 2X and the least amount in the baseline, but we also suffered with the 3X in terms of tannin synthesis. And what was really interesting is that we saw this, so this was due to heat wave 1. So clearly we're having an effect of these early heat waves on tannin synthesis, which occurs much earlier in development. And so I think that actually has surprised a lot of people, but that was really interesting to us. And we're doing a lot more work in terms of doing some of the transcriptomics and more chemistry to understand that impact. And also too, because some of those chemicals are supposed to be more heat tolerant, some of that chemistry. So, and lastly, I'm just gonna briefly show you, I'm not the one to dive into this in detail by any means, but we did do an analysis on the wine after 3 months. And interestingly, and this is mostly parameters that are related to, we didn't find tons of interesting data on the phenolics. I'm only showing you what's significant. And this is just a PCA, a pretty basic analysis of clustering. And we do find that the wines cluster. So this is having an effect on the actual wine chemistry too. And this is only after 3 months post-bottling. And we were supposed to do a sensory study, but of course, that's not, who knows? This might be like a very well-aged wine by the time we do a sensory study. So just to close, I was actually very surprised that we always saw a pretty strong recovery from after the heat waves due to the physiological stress that was incurred. And we're actually finding some similar responses this year, although we looked at some of the data from yesterday. We looked at it this morning, and there is not, for some reason, there's a little bit less of a response this year in terms of them separating out in terms of stress. But that also probably is, I don't know, it could be due to some of the other irrigation decisions made this year, but it'll be interesting to see what effects there are on berry chemistry. And again, as I mentioned, the main influence we saw that was lasting was this influence on berry composition, yield, and some of the wine properties. And we also found that generally negative results were observed from excess water as well as from insufficient water application. So our goal longer term, working with the Macaron Lab and others doing some modeling, is to actually hone in on what you want, what physiological parameters are the most relevant to be able to make decisions about irrigation, and actually develop guidelines that are less sensitive to a specific region, and say, hey, we think stomatal conductance, and other work has found this too, is actually a really nice target for irrigation. And some growers are doing this now. And so that's what we're working on now, is actually having continuous monitoring and doing remote sensing and some hyperspectral sensing as well, to figure out what the most relevant parameters are to help guide irrigation, and also do it in a way where it's not an expensive endeavor, where it's something that's accessible to growers and can be used easily. And so with that, I would just like to thank my lab and collaborators and all of our funders, and especially Gallo, who's been amazing as far as helping us make this happen. And then 2, we're going to be starting doing experimental trials here at Davis, and then potentially at Napa. So getting some feedback from all of you in terms of what you think is the most relevant, and in terms of thinking about the overwatering, because we're starting to think that perhaps these roots, that they're anoxic conditions that are created when you overwater, and that could be causing some damage and suffering. And lastly, I'm going to continue to plug our survey, and I will put it in the chat. And then also too, if there is anything, feel free to reach out to me. You can either email me or DM me. I'm young and savvy. So yes, thank you so much again. And I hope to come in person and present and talk to all of you before too long.
Thank you very much. So after the first 3 speakers, does anyone have any questions? You can ask in the chat, you can raise your hand, you can unmute and just ask. Anyone? Well,
this is Stephanie Karen.
Sure.
I just wanted to say thank you. It's so nice to have some good energy into the department, and we're very excited to help you guys as much as we can connect with Lodi growers, and just please stay in touch with us. I know this is a weird time, but we really appreciate all of the enthusiasm, and we look forward to working with all of you guys as you launch these great careers even further. Thanks.
Well, thank you, Stephanie. So Stephanie is a good jumping point as well. And I should say that we've been working pretty closely with the folks at the Lodi Wine Grape Commission, and we really appreciate them helping to get the word out. So you can either go through them to get in touch with anyone here at UC Davis, or myself, or Dave Block, or Anita, or anyone that you see here today. We're pretty all easily accessible on the UC Davis website. Oh, there's a question for Mason here. What are the kinds of sensors you're testing for your low-cost system?
Thanks, Donnell. So as far as the low-cost sensing system that we're working on, right now we're really focused mostly on computer vision-based ones, so image-based sensing. So the goal is to keep it what we consider to be relatively low cost. So for the whole thing, for the processing, the sensors and everything, somewhere between $1,000 and $2,000. And so they are, right now it is red, green, blue. So that's your standard camera image. It's an infrared. camera, and then also a depth camera, which tells you how far away different things are. So we're trying to combine those three for the detection and prediction of things like phenological states. So that's something we're working on with Beth, where we can try to understand early on what the phenology is and use that as part of the model to predict both yield and when time to ripeness might be. That's a big part of what we're focused on now is vision-based imaging. But I think there's a lot of opportunity for, I guess we are doing a little bit, also Dave Block and I are doing a bit of work on developing sensors related to water stress and predicting evapotranspiration in kind of a low-cost sensing type of system again as well. So those are the two main ones right now, but looking forward to growing the lab and kind of finding additional applications and looking at additional sensors as well.
Well, thank you.
Thanks.
All right. Anita, are you ready to share your screen?
Yes, let me just. Now I just need to find my mouse. Share screen. Okay, I have too many things open. I can't even find the screen I'm looking for. Just give me a moment. Sorry. Let me just close like everything else that I have open. Other than Zoom. That would not be a good idea. Okay, let me try this again. Share screen. Oh, there you are.
Aha, there you go.
Ah, there we go. This will work. No, I have like 20 things open. I'm sorry.
That's good. That worked well.
Good afternoon, everyone. I just finished my Red Bull, so I should be ready to go. I was asked by Karen to talk about some work we did in collaboration with E&J Gallo for many years. And I'm saying Anita Oberholster and David Block because this really was a collaboration between the Oberholster and Block lab. And I'm going to talk about fermentation parameters and their effects on wine quality. So first, I'm going to talk a little bit about the motivation. Why do we need to look at, cap management, for instance, parameters 0 in wine fermentations and the impact that has on the extraction of phenolics, the key compounds that we focused on in wine. And then what parameters were actually investigated. We looked at cap and liquid temperature, pump over volumes, pump over frequency, cold soak, different cold soak durations. And then we went into the more basic chemistry to try and explain what we saw, looking at the absorption and desorption kinetics onto solids in your fermenter. And then the modeling part, I'm going to leave to the engineer, which is Block, not me. So, okay, so this is also a slide that I stole from David Block, so just in case you see it again. So basically, we know during fermentation, we have a dense cap forming at the top of the liquid below. And this is because we have the release of carbon dioxide during fermentation. It pushes the polymers to rise, and we know that we get temperature for differentials between the cap and the liquid below, because it's pretty easy to remove the heat from the liquid below just with jacketed fermenters, but it's not easy to remove the heat from the cap because it is a poor conductor of heat. To do that, you really need good mixing. So Dave Block's earlier work have shown that you can have up to 12 degrees C difference between the cap and the juice. Now, why is it important to remove these differentials? Well, you want uniform fermentations. You don't want to inactivate a subportion of your yeast, but also cap management is important for phenolic extraction. Okay. Color, monomeric phenols, tannin, polymeric pigments. We know all of these are very important for red wine quality. So what we did is, if you look at anthocyanins and flavonols, they're in the skins. And we analyzed the flavanol quercetin and molybdenum-3 glucoside as basically a marker for skin extraction. Then if you look at tannins, you have their building blocks, of which catechin is one. And then you have various proanthocyanins. Now, they're both in seeds and skins, but we specifically focus on catechin as a marker of seed tannin extraction because there's so much more in the seeds. And then we look at hydroxycinnamates because they're mostly in the pulp. We saw that, looked at that as a marker of pulp extraction. Now, because I'm limited in time and you don't have an hour, I'm really just going to show you the highlights focusing on anthocyanins and catechin. So this is the molecular mechanism for cap extraction. Another thing that I stole from Dave Block. See, I do that a lot. So basically what happens is your phenolics are present in the vacuoles of the hypodermal cells, their skin phenolics. And when you crush a fruit during fermentation, they get released. So that's the release portion, you see that. And after they get released, and obviously some of them actually get extracted through the cell wall membrane as well. After they're released in the liquid, many different things can happen to them. One is degradation. The other is secondary reactions to form polymeric pigments and co-pigments and other things, or reabsorption. We know that reabsorption do happen onto grape solids in the fermenter as well as yeast. So the first thing we did is we wanted to know, so that earlier work showed that you get temperature gradients in the fermenter. So the next step was, okay, do we see phenolic concentration gradients in fermenter during extraction? So we did everything I'm discussing today on Cabernet Sauvignon. And we did this 2 years in a row on the same fruit in a 2,000 liter fermenter. And we put down this curtain of temperature sensors, but we also added these 15 basically sampling boards. And we took samples from them at 5 different levels, as you can see on the right. So this is a picture of what that sampling port looked like. And so our other thing is, the first row, very quickly we realized after like a few hours, we can't get any samples from the top layer of the cap because it's just too dry. So we excluded from all the rest of the calculations, we excluded the first row that you're seeing there. But so you basically use a peristaltic pump on the catwalk to pull samples at specific levels in the fermenter. Now, what you're seeing here is we constructed these contour plots based on the 12 sampling ports you see there in the fermenter. Okay. And this is for every 24 hours. We did pump overs twice a day at 9 o'clock in the morning and at 5 p.m. in the evening. And we took a sample just before a pump over, so that you can see the concentration gradients forming. And what you can see here for the case of anthocyanins, you can clearly see the gradient forming in the cap versus the bottom. And as you do a pump over, the liquid below, also the concentrations there are also increasing. And you can see by day 5, after day 5, you don't see any concentration gradients between the cap and the liquid. It's because your extraction has been complete. And we've seen this, many other researchers have seen this, that in a fermentation temperature of around 25 degrees or more, your anthocyanins extract really quickly. And this graph at the bottom basically shows you the 2 top lines are the 2 top layers, bottom ones are the bottom layers that's in the liquid versus the cap, and you can see after about 5-6 days, they're all come together because you don't see any extraction from the cap anymore. Now, if you basically reach an equilibrium, that doesn't mean there aren't any anthocyanins left in the skins, obviously. Now, if you look at this, this is basically an hourly. So you can see how that differential, concentration differential actually developed. The 8 a.m. just before the pump over on the 3rd day of fermentation. Then you can see 10, 11, 12, 1, 2, 3, 4 p.m. You can see that differential just before we do a pump over again. When we did the same thing on day 5, we basically saw nothing because there was not much extraction happening from the cap anymore. I'm not showing you the tannin data, but the tannin data basically, you just saw an increase in tannin extraction until the end of fermentation from the cap. And we've seen this, people that's done extended maceration, sometimes you can see extraction until 45 days because there's so much there at the source. And it takes a lot for that to be saturated and reach an equilibrium. I always forget my animation, I'm sorry. So the next thing we did is we looked at, okay, so we know there's concentration gradients forming. We know we did an R fermenter one pump over, one volume, twice a day, and it worked really well to remove these concentration gradients and temperature gradients. But then we wanted to know, okay, what is the impact if you have a differential between a cap and the liquid? So we did this experiment. We did 20, 25, 30, and 35 degree fermentations, where we kept the cap and the liquid below at the same temperature, and when we tried to maintain a differential. And we determined the most easiest differential to maintain would be around 5 degrees. So it's 20, 25 in the cap, 25 in the liquid, 30 in the cap, 30 in the liquid, 35 in the cap. So we did this in our research fermenters. We used the integrated fermentation control systems, or IFCS units, much easier to do this. So basically we had 3 temperature probes, one in the jacket, and that jacket temperature was at the temperature we wanted our must at, or our liquid portion. We had a mass temperature probe and we had a cap temperature probe. So when we wanted them to be at the same temperature, we told the IFCS unit, if the difference between the cap and the mass is more than 1 degree, do a pump over until they're the same. So it basically means that there was a lot of pump overs going on. Luckily, it was automated. When we wanted differential between the cap and the mass of 5 degrees, we told the IFCS unit, if the temperature differential is more than 6 degrees, do a pump over until it's a 5 degrees difference again. Now, if you look here, we also do have automated Brix measurement and temperature measurements, and it constructs these fermentation graphs or progress curves for us. So what you can see here, this is Brix. So this is you can see the progress of fermentation. But obviously this was a trial where we did mass temperature of 25 and cap temperature of 30. And the interesting thing here to note is that we could only maintain that temperature difference for about 3 days during the active portion of fermentation. So let's have a look at what Malverdin and catechin extraction look like for these different trials. Now, just to make it easy, I'm just showing you the data for where the mass and the cap temperature was the same. The most interesting thing here is for 20, 25, and 30, there really was not a significant difference in extraction of Malverdin-free glucoside. However, you can see at 35, there was significantly less by the end of fermentation. We believe this is because extraction happened really quickly and we got the same amount as the rest and we saw this sudden decrease. Yes, some of this is due to polymeric pigment formation, but not enough of it. We really do feel strongly that we saw deterioration or degradation of Malverdin-free glucoside happening at 35 and above temperatures. So that's something to be aware of. If you look at catechin concentration, not surprisingly, you can see an increase in extraction at higher temperatures because they're more soluble at higher temperatures, and that is maintained. If you look at the next graph, so next question is, okay, so what was the driver for extraction? Cap or mass temperature? So this is only the mass temperature. And if I overlay the trials where the cap and the mass temperature were different to each other, is really busy. But believe me when I say the 2020 and the 2025 cap bicyclize on top of each other. The same for the trial where we did 25 cap and mass temperature, and we compare that with 25 mass and 30 degrees in the cap. And this makes sense because we could only maintain this differential in temperature for a short period of time, and it just make up for this difference. So really the main driver is mass temperature for extraction. Now, obviously, while we were doing this, we were doing a lot of different frequencies of pump overs and total volumes of pump overs. And people say, well, you know, and we thought the same thing. This could influence what we saw, could influence the phenolic extraction. So we did a lot of different tests on the same grapes in different years. So first we did the graph that you see that there is for Malverdin and tannin concentration, doing a total of 4 volumes, which I know is a lot. Doing them at, and now I can't see my own screen, I think it is 1 times, twice, 4 times, and 8 times with a total of 4 volumes. And we saw no impact whatsoever. We did that because we did a lot of pump overs during that experiment. Then we also did a test which was a little bit more comparable to what most people may do in their fermentations. We did twice half a volume, twice 1 volume, and twice 2 volume, and saw no impact. The graphs really looks exactly like the ones you see here on the right. But then people said, yeah, but perhaps that just wasn't extreme enough. So we went more extreme. We went half a volume. We went half a volume twice a day, half a volume once a day, quarter of a volume twice a day, and no pump over. Now, there really wasn't a significant impact. Now, you have to remember, we did this in our research fermenters. So there are about 50-gallon fermenters that we fill probably about 30 gallons of. So there's a lot of natural mixing that will happen as well because of the release of carbon dioxide. And in our fermenters, even a no pump over fermenter didn't make a huge impact. But that doesn't mean, obviously, that we're recommending that somebody with a 30,000 gallon tank go and do no pump overs. What it did say for us is if we do 1 volume twice a day, we probably get something good, and we can probably get away with even half or less of that for our fermenters specifically. Then we looked at the question of cold soak. So we did this for 2 years. The first year we did it on Cabernet Sauvignon from Lodi, and the second year we did it on Cabernet Sauvignon from Paso Robles. Now, we did this on purpose. We wanted Cabernet with different phenolic content. So the ones in Paso Robles had about 30% more color, 30% more tannin. Also in the second year, we add one variable. We added 2 days of cold soak because that was the commercial practice of the winery we compared our data to. Because in the second year, those trials that's in the red blocks, those were actually repeated on the same grapes in 30,000 gallon tanks. And they found comparable results to what we found. So what's the theory behind cold soak? The theory behind cold soak is that it's an aqueous environment, so you have no alcohol. So this will increase in extraction of your hydrophilic compounds, which are compounds like your pigments, because they're glycosylate, okay? They have sugars attached to them, very soluble in water. Then the idea is also you don't have a cap yet. So your skin gets broken down and there's better contact and you get more extraction of your skin tannins, which everybody wants. But now this doesn't gel 100% with what we know chemistry-wise happens. We know both tannins and anthocyanins are actually more soluble at higher temperatures and at higher percentage of alcohol. And we also know that you can get seed tannin extraction even in an aqueous environment. It's pretty slow in the beginning because the seed needs to get dehydrated, but after that, you do actually get extraction. So you do need to be careful, even during cold soak, of how much seed extraction you can get. Now, this is a pretty busy graph, I apologize, but if you see that dotted lines, that's where the cold soak stops. 1 day, 4 days, 7 days, and 10 days. And basically what you want to look at is, if you look at the end, they all seem very similar. Now look at the bottom graph on the left. That's where all of these different cold soak durations, what their anthocyanin concentrations were at the beginning of fermentation. And you can see the longer the cold soak period, the more anthocyanins they had at the start. But by the end of fermentation, they were all the same. And this persisted during bottle aging. So it had no long-term impact on anthocyanin extraction. And we saw the same thing for the next year in different set of grapes. If you look at tannin extraction, you will see also the longer the cold soak duration, the more tannin extraction you had. And that was maintained during fermentation. So you actually did have more tannin. Now, in the case that I'm showing here, it was really only significant between 0 and 1 days and 4, 7, and 10 days. And the second year, it wasn't significant. It was just a trend. We did also see a trend in seed tannin extraction. It wasn't yet significant, but we could see seed tannin extraction increasing over time. So sorry, somebody's ringing me. Okay, so the next thing we did is we also did sensory analysis in the second year, and mostly it was a small difference in like caramelized sugar on the nose, but it wasn't significant yet, but there was an increase in bitterness perception for 7 to 10 days. So it's just something to be aware of if you're interested in doing more longer term cold soaks. So what did we learn? Cap management conclusions. cap management is really important to remove accumulated heat from the cap. It prevents heat inhibition in the cap and removes extraction saturations close to the cap. We do know that if you couldn't do pump overs, that you can make up some of that loss later on. If it didn't overheat and didn't kill your yeast and degraded your anthocyanins, you can actually make up that extraction afterwards. We also know that 1 to 2 volumes twice a day are probably adequate for most fermentation tanks with a good aspect ratio. Very complicated regimes probably doesn't have as much as an impact as people may think, but you need to figure out what is the optimum to remove concentration gradients and temperature gradients for the specific fermenters you are working with, definitely. And for us, in our case, cold soak didn't have a lasting impact. But I have to say, because a lot of people having done cold soak studies find different things, I've looked to see, okay, what's the difference? We followed our cold soak with what we call a pretty warm fermentation, 25-degree jacket. So that temperature went up to 28 probably, if not 30 degrees Celsius, or now I'm talking around 85 degrees Fahrenheit. So basically what that means is we followed a cold soak with hot fermentation of 7 days. If you were gonna press after 3 days, or you're doing a cool fermentation, you may be seeing something different. Perhaps that difference is actually maintained for you as far as color goes. Just something to keep in mind. So after we've gotten all this, so the idea is really that if we can predict phenolic extraction, that we could model this so that we can look into, okay, what's going to be your fermentation conditions? What is your starting grape phenolics composition? Can we predict at what extractability you're going to get? What's going to be the phenolic composition of your final wine? And then you can work it back. You can tweak your fermentations so you get what you want and what you're aiming for depending on the grape composition of that year. But we didn't have enough information yet because we mostly worked at this point on release and the impact of fermentation parameters on that release from the skins and the seeds and the pulp. The next thing we wanted to focus on was on the absorption, desorption kinetics. Now, I'm not going to spend a lot of time on this. This is like the last minute, I promise. So basically, because I'm looking at Karen. So what we did basically is we isolated pure anthocyanins, we isolated skin and seed tannins, we isolated cell wall material. That was a lot of fun. And they, not really, but my students did it. And then basically what we did is we combined them with cell walls separately and together at different conditions that you may expose these compounds to during fermentation, 15, 22.5, and 30 degrees Celsius, 0, 7.5, and 15% alcohol. And what we found, I'm going to summarize in one slide, 3 years of work. So anthocyanins absorb faster than grape skin and seed tannins. Absorption decreased with an increase in temperature and alcohol, which is good news. During fermentation, less and less, or what have absorbed can desorb again because both anthocyanin and tannin absorption is reversible. This is coulombic interaction, hydrophobic interactions, etc. These are not covalent bonds. And we also saw a preferential absorption of larger tannin, which is less good news because we want those larger tannins because they're softer and they're less bitter. And we saw this is enhanced in the presence of both anthocyanins and tannins. We think this is partly due to polymeric pigment formation and binding. We don't know if it is because pigments bind to smaller tannins, so the bigger ones are available for absorption, or whether it is because the pigments are binding to the larger tannin and preferentially binding to the cell wall. This is something we are trying to figure out at the moment. So basically, all that data was used by Dave to do the things that he's going to talk about that is way more interesting than what I just told you. This is a list of many students and other people that's collaborated on this project up to this point. And obviously, these are some photos of my lab. And I have to thank E&J Gallo Winery because most of the funding did come from them. Thank you very much. And now I'm going to stop sharing my screen before I'm in trouble.
Thank you, Anita. So we'll move along to Dave Block, and he will do his own introduction so we can keep things moving quickly.
Can you see that? All right, can everybody hear me okay? All right. So as we're running a little bit over, I told Karen if she wanted to skip me, she could, but she said, no, I can have 3 minutes to give my whole talk. And I said, how about 5? And she said, okay, 5. So we'll see. I'll try and give you kind of the gist of what we're doing that Anita already introduced. So Anita already talked about all the observations we made on phenolic extraction. And the next thing we wanted to do in our labs was to see if we could use that information to generate a model that would let us predict what was going to go on for phenolic extraction in a red wine fermentation. And if you have a model that's predictive, then you can use it to understand what's going on, and you can also use it to optimize what you're doing and try and get the phenolic profile that you're shooting for. Almost all of this work that I'm going to talk about was done by a PhD student that worked with me and Anita. His name is Conrad Miller, and he just finished recently. So we'll talk a little bit about the problem, but Anita already introduced that. And then I'll talk a little bit about what we've done to predict temperature in red wine fermentations, because it's not uniform, as Anita already said. And then modeling phenolic extraction. And then quickly, actually, we'll spend a few minutes talking about some case studies to illustrate how you can use this model that we developed. So I think the key point that Anita already really described is that phenolic extraction red wine fermentations is really complicated. And so we understand some of the results we got, but as Anita said, things like why doesn't the pump over frequency or volume make a difference? We didn't really understand that. And so in order to understand this better, what we decided to come up with a mathematical model for phenolic extraction. So physical and mathematical model. So what does that mean? So the first model that we developed was a model to predict, as a function of where you are in the fermenter and time, how do temperature and ethanol concentration vary in that fermenter over time. And we wanted to do that because we know that phenolic extraction is a function of temperature and ethanol concentration. So this first step is, how do those vary over time and space in the fermenter? So to do that, the model has several parts. The first part of that model is a model for yeast cell growth. This is a model that we developed about 20 years ago in my lab, and it works pretty well to predict yeast growth as a function of initial nitrogen concentration and other factors. Next thing we need is the physics of what's going on in that fermentation. So we had equations for that. This first one is mass transfer of those molecules that Anita talked about into the liquid from the skins. The second one is fluid flow, the flow that you get from things like temperature gradients in the lower part of the fermentation, which is well mixed, as opposed to the cap, which is not mixed at all. And then this last equation is heat transfer. So we know that the liquid in the bottom is cooled pretty well, whereas the cap is not really cooled at all. There's not really any heat transfer that cools the cap down appreciably in a red wine fermenter. And then the third part is physical parameters that nobody had actually measured. So Conrad went into the winery and actually measured things like what's the thermal conductivity of pumice over the course of the fermentation. So with those, we could actually predict temperature profiles really well in a fermentation over time. We had collected data, as Anita said. We compared it to that, and it compares really well. I was going to show you that data, but I'm going to skip over it, and you're going to have to take my word for it that the fermentation model, this model works really well. All right, so the next step is to take that part of the model and to actually add to that the phenolic dynamics. So Anita showed you this already. Phenolics are released from skins and seeds. They react and they absorb and desorb. And so what we wanted to do was to come up with a mathematical model for that part. And so that's the next step, and to have this model for phenolic extraction as a function of temperature and ethanol concentration. So the model that Conrad developed looks like this. The details of it aren't hugely important for you, but we have a mass balance. We have equation describing the release of all these components from the skins and seeds. We have reactions that include, for instance, the degradation of the anthocyanins that we witnessed in our experiments. And then we have the adsorption and desorption equilibrium. We have an equation for that. And the constants are ones that Anita's group actually measured in the last part of her talk. So it turns out when you use those equations, and I'm not going to show you this data, you're going to have to take my word for this too. But when you use those equations, it models what happens in a very small fermentation, a very small well-mixed fermentation, really well. And so we can take these two parts of the model, the phenolic part and the temperature and ethanol part, and actually put them together into one model and see if we can predict what's going on in the actual fermentation. So this is data like Anita showed you. I think she showed you anthocyanin data, and she said she wasn't going to show you the tannin data, so I decided to show you the tannin data. This is what it looks like. This is the experimental data. And then what the entire model shows you, this is the predictions of the same data. And it's not perfect, but it's actually really good considering that we really didn't make any assumptions. We just used the underlying physics and the parameters that we measured, and it actually works pretty well. We have a lot more data that shows that the model predicts well, and that I'm not going to have time to show you, but this gives you an idea that we in fact prove that the model predicts what's going on in a red wine fermenter at different scales really well. How do we use this? This is probably the most important part of the talk to you, is if you trust me that the model works pretty well, then the key part is how do we actually use it to, how would you use it as a winemaker? And I liken this to a GPS system. So basically what the model does is if you actually put in where you're starting, which are your grape characteristics, and you put in where you want to end up, which is your desired phenolic profile, then like a GPS on your phone, it will tell you what's the route to get there. What should you do in terms of pump overs, fermentation temperature, and so forth to get there. And there may be multiple routes that it could suggest, and you can pick the best one to get to the phenolic profile that you're shooting for. So as an illustration of that, we use the model in 3 case studies to illustrate that point. So in this first one, we're simulating a 2,000 gallon fermenter, so probably small on a lot of your scales, but reasonable industrial scale. And what we found was if we wanted to maximize the tannin coming just from the skin, we could do that by having a pump over once per day. If we actually didn't pump over at all, or we pumped over 8 times per day, we actually saw lower skin tannin. And this is a balance between the temperature, because the more you pump over, you get a lower temperature, and this kind of fresh solvent that gives you more of a driving force for extracting the skin tannins from the skins. And that's not an obvious result, so it's something that the model can help you decide. Second case study is, what if you wanted to minimize seed tannin, what would you do? And at both a 25 gallon scale, which is kind of what that TJ scale that Anita talked about that we use in our winery, and a 2,000 gallon scale, the more you pump over, the more you reduce seed tannin extraction. And this is because essentially you're reducing the temperature, and seed tannin extraction, as Anita told you, turns out to be very dependent on temperature, more so than skin tannin extraction. And as a third case study, Anita mentioned that pump overs didn't seem to have much effect on extraction at the scale we're working at, and we didn't really understand why. And so we used the model to try and understand why, and it turns out the model tells us that it's not, that what's really critical is not the volume that you have, but the distance between the bottom of the tank and the bottom of the cap. And so the shorter, wider tank you have, the shorter that distance, and the less that pump overs will make a difference. If you have a tall, skinny tank, pump overs are going to make much more of a difference. And we've seen even commercial scale winemakers tell us they can actually not pump over and get good extraction. It's probably because they have a short, wide tank. All right, so that's just to summarize. We basically took all those observations and we put them into a mathematical model that predicts really well. And what that mathematical model can do for us is to actually allow us to optimize, use as a tool to optimize phenolic profiles for winemakers. So that's what we're working on. We're working on making the analysis, the chemical analysis faster and the model easier for anybody to use. And once we have those, it could be a very interesting tool for winemakers to use to optimize phenolic extraction in red wines. And so I'll just finish by acknowledging all the people who did the work, and especially American Vineyard Foundation that funded all of this work. And so with that, I'll turn it back over to Karen.
Thank you, Dave. And next up is Ben Montpetit. He is now, actually I had on this particular agenda that he was an assistant professor, but as of July 1, he is an associate professor in the Department of Viticulture and Enology at UC Davis. And he's going to be talking about site-specific factors and Pinot Noir fermentation outcomes.
All right, thanks, Karen. You can see the slide there, no problem?
Yep.
And my mouse, you can see on top of that. Moving around a little bit. Excellent. So yeah, so I'll take the opportunity to give you a shortened version of what I was envisioning for today, just to keep everyone on close to time as possible. And in large part, what we're interested in here is looking at and deconstructing, various fermentation outcomes. And I take a very much a yeast-centric point of view to this, and you'll see what I mean by that as we go forward. So the work I'm going to talk about today piggybacks on a larger project that Dr. Runbaum has been running at UC Davis, which we refer to as the Pinot Noir project. And this basically has the kind of overarching goal to try to understand the impact of site on the sensory and chemical characteristics of the resulting wine. And you can see on this map on the left, the sites that we're referring to and the kind of geographical distances that we're covering. And what's, I think, unique about this project and nice about the project is material from all these vineyards comes to UC Davis, and it's made we do quadruplicate fermentations. So in these TJ fermenters, highly controlled fermentations. And in that way, we think the kind of variability that we're seeing fermentation is minimized, and this obviously aids with the data that we're able to extract. So when you're thinking about the impact of site and how this impacts fermentation outcomes, one of the first things you may start to think about is the weather that these sites are experiencing and how this impacts development of the plant materia and also the associated microbes that come in with the grape material. So this is one of the first things we thought about as well. And we all know that a fermentation is a result of a very complex mixture of microbes. And this is just illustrated in this set of graphs that are taken from one of David Mills' publications. And again, for the sake of time, I won't go into it greatly, but you can tell that over the course of these fermentations, from must all the way to wine, things are changing a lot. And we have technology that we can use to follow these microbes over time. And we've used some of that technology here to look at the various sites and the fermentations and what microbes are active in these fermentations. And there are basically two different technologies that one could employ to ask this question. Most of the commercial technology that's available to any of you, for instance, if you wanted to get some analysis done on your must, or is based on the analysis of DNA, so going after the genetic material that's present, and by knowing what the genetic material is, we can correlate that with a microbe. Importantly, DNA is a very stable material. It's present in cells that are alive or dead. Active, inactive, doesn't matter, it's present. So really the signal that you get from DNA based technologies is telling you about the potential of a site, that these microbes are there and they could potentially be active. And when we do that and we look across sites, we of course find hundreds of different species of microbe that are around bacterial and yeast. But one thing we could do is look across these sites and ask, are there unique organisms on any of the sites? And to make a relatively long story short, the answer of course is yes. And we can actually find that based on looking at these unique microbes that are on each of these sites, that we can predict with about 95% accuracy what vineyard is which based on the microbes that we detect in these fermentations. So clearly there are site-specific microbial populations. To give you kind of one snapshot of this, this is a graph just showing across sites the presence of this one lactobacillus bacteria. And you can see here there are only 2 sites that this microbe shows up on. And these happen to be both sites that are in the Santa Maria Valley. So it's telling us again that these sites are unique, and they're unique at the level of individual species of microbe that are there. Now, remember I said that this speaks to potential. So this tells you that those microbes are there and could potentially be contributing to an outcome. So a second technology we deploy is one based on detecting RNA that is present. So RNA is very similar to DNA. It's actually copied from the DNA, but the important thing about it is that it's fairly labile. It's easily destroyed, and in that way, it's only present in cells that are living and active. So once cells die or become inactive, the RNA signature largely disappears. So we think that by measuring this, we're actually measuring what is actually present and active at a given moment in time. This technology is really research-based. It's a little bit prohibitive based on cost, and it takes a little bit of a different expertise than you might find in a winery lab. Not saying it can't be done, but it would definitely require specialized training. And when we do this, we see a different signature. So what I'm showing you here is data taken based on this RNA signature, again, this active population of cells at 16 hours post-inoculation. So this is after cold soak, and then the fermentations are warmed up, yeast are inoculated, and this is 16 hours later. And you can see here in the vast majority of sites now, Saccharomyces cerevisiae, for instance, is the dominant microbe, except for on 2 sites. 2 sites show a very different profile, and you actually see that non-Saccharomyces yeasts are the dominant species that are present and active. And Saccharomyces is definitely there, but is not the same as these other sites. So what this is telling us is the information we're getting from looking at the potential, the DNA signature, and what's actually active is different. And I'm just putting these 2 side by side to give you an essence of that. So on the previous slide, you may have noticed Uvarum, Hanseníaspora uvarum, as one of those species that was present and active in the Santa Maria Valley sites. That's represented here. But here, this is actually the signal that comes from the DNA information. So if I thought that there was something unique about the Santa Maria Valley sites and was interested in investigating what that may be, and I use this information here, I would say that, this can't be Uvarum that's contributing to this uniqueness because it's present on all the sites. But what this information tells us is, although it's present on many of the sites, it's really only active on these 2 sites from the Santa Maria Valley. So we think that this information is adding quite a bit to our understanding, and we'll be able to pinpoint differences. Going further though, of course, there are many other reasons that a site may be different. Maybe it has to do with metals and in the soil and that come in with the grapes, and other developmental characteristics of the grape material. And of course, then when the wine is made, you have many other variables like the winery and the person making and overseeing. So again, with the Pinot project, what we're trying to do, or what I've kind of come up with as an approach, is to try to turn this and ask, can we use Saccharomyces cerevisiae in a sense can we use it as a sensor to tell what is going on? And the reason I want to ask this question in this way is, again, I'm a yeast biologist by training. We know a lot about this organism, and it turns out with the technologies we have, we can follow in real time what it is doing. And in that way, it could actually be reporting on the inputs that it's receiving from its environment and the signals, and then that will tell us about what it's actually doing. So this is a Another diagrammatic way of saying this is that we think Cerevisiae is obviously a very complex biosensor. It's going to sense its environment, it's going to process that information, and it's going to respond. And if we can monitor that response, we learn something about the environment. In the context of the Pinot project, we eliminate some of the variables again. You know, the wine's all made at UC Davis by the same winemaker. It's all in the same facility. So again, we think we're going to pull out traits that are really specific to the microbes and the grapes that are coming to UC Davis, and will speak to site. So the approach is fairly straightforward. We sample fermentations 5 points across the fermentation, and when we extract this information about what Saccharomyces is doing at each of these points, and we use our knowledge of the system of Saccharomyces to try to interpret, you know, that in the context of the fermentation. The pieces of data that we're following are what we refer to as gene expression data. So we monitor what genes are on or off at any given point in time. Saccharomyces has about 6,000 genes. So we're monitoring 15 different sites, fermentations from 15 different sites, 5 time points. We're doing it in duplicate across 6,000 genes. So we're analyzing a lot of data, and we've done this now in 2017 and 2019. So I'm not going to obviously go through such kind of an amount of data, but what we've been able to do is distill each of these time points down to a single kind of spot on a graph, and that's represented here. So this is what we refer to as a diffusion map, and what you basically need to know about it is it's color-coded based on Brix, and each of these points represents a sample that was taken from a fermentation. And you can't tell what vineyards are which on here. I would have to color code that differently, but hopefully what you can see is the way this diffusion map separates the data. Things early in fermentation are here on the left. These are high in Brix values, and then over time they separate and they move down here, and then they spread on this component too. Now this is, you know, maybe interesting to look at, but what's more important about this is that we can then ask of the data what is causing these separations on these different components. We can ask what the drivers of this are. So when we go into the data and ask this question, on this axis, component 1 causing this shift that you're seeing here, not surprisingly, this has to do with macronutrient availability. The genes that are responding and causing these changes have to do with sugar metabolism, nitrogen metabolism, and you're going from environments where these compounds are very abundant to low starvation conditions, and this drives this kind of distribution that you're seeing. But what we can then do is query the data in the other dimensions, and we can, for instance, look at this dimension and say, although these are all late samples, they're not clustering like these early Brix samples. They're actually separating over quite a distance here. So what is the driver of that? And I'm just giving you one example. There are actually many components to this, and we're still working to figure them out. But in both the 2017 and 2019 data, one of the biggest signatures that we get is around the nutrient or vitamin thymine. And in the context of a fermentation, it's always nice to see compounds that you know already that are important. You know, so just to give you an example, thymine is a key to making some cofactors that the yeast use to drive metabolism. And for instance, here is one place this cofactor is used, which is a very well-known pathway amongst all of us, which is how the yeast is making ethanol. So again, it's, you know, it's kind of interesting to see this compound come out, and it gives us some information about what may be differentiating these sites and where these fermentations end up. For those of you who have an eagle eye and maybe can see this, hopefully it's not too small and you might have seen this, is that there are a few points over here that look quite different. Most of the yellow ones are on this side, but these two are clustering way up here and way over here to the right. So it suggests to us that these sites are very different from the rest of these sites. And based on some of my comments already, you may have a guess at what this site is, but this turns out to be the Santa Maria Valley sites. So if you remember, these are the sites that from a microbial perspective are quite unique. They have different activities than these other sites, different microbes present. And this suggests to us that there might be something very microbially driven that is driving these differences. And it suggests that Saccharomyces is changing its metabolism based on the presence possibly of these other microbes. This is not a new concept. You can find publications. This is one that looks at yeast interactions, but there's also yeast bacterial interactions. We would refer to these as interspecies interactions, where it's known that the presence of one yeast, for instance, can impact another yeast and alter the way it behaves. This could be synergistic and positive. It could be negative, where they're actually competing and trying to harm one another. And this often goes through the production of antimicrobial compounds, competition for nutrients, etc. So we envision a scenario where these fermentations and where they're ending up in multiple vintages may be driven by unique microbes that are present in the Santa Maria Valley, for instance, and this is a driver of these fermentation outcomes. So we think that this kind of pilot study is indicating to us that we do have the ability to use Saccharomyces as a sensor and to follow these fermentations over time and extract important drivers of these fermentations and their outcomes. And I think what's important for all of you, for me, the biology here is very interesting, and I can get very caught up in a few genes or some pathways that are being manipulated, et cetera. But I think what is important is, based on these signatures, we can develop, kind of actionable information, which tells us that, for instance, these Santa Maria Valley sites, for instance, have unique microbial populations that are linked to unique outcomes, and that there are specific nutrients that are driving these patterns of change, which then could be targeted. Thymine is actually a component of a lot of the nutrient additions that are made, some of the components you might use to start fermentations, etc. So, if you know about these nutrient imbalances or these deficiencies, is that something that could then be actionable and could be actually added to a fermentation if allowed to alter an outcome. Okay, so with that, just stop, acknowledge the people who did this. Most importantly, Rachel and Taylor are the people behind the scenes generating all the data while I sit in my office and bang on a keyboard. And of course, those people who make it possible with funding. And with that, I will stop, and hopefully, for time, I didn't go too fast, but I'd obviously love to take any questions if anybody has any questions to close out.
So if anyone does have any questions, again, you can either raise your hand, you can just unmute and ask. You can type it in the chat. While you're thinking about your questions, I'd like to thank all of the speakers for taking the time to speak to everyone today. And again, I want to thank Stephanie Bolton at the Lodi Wine Grape Commission and Amy Blagg at the Lodi District Grape Growers for helping us get the word out. And, we have an ongoing dialogue, and anytime anyone has any questions, you can either go through them or just go directly to us. Either way, whatever's easiest for you guys. But just so you know that we're all trying to work together to make sure everyone is successful. So with that, I'll take any questions.
For anybody?
Yep, for any of the speakers, if there are any. Anyone?
They're shy. It's because it's not office hours and me and Dave aren't asking questions.
Hey, there's a question from Donnell who asked, are those records behind you?
Yeah, so not what I was going for in this presentation. I obviously had a question about my Zoom background, but nonetheless, I can answer it. This is the KDVS studio here at UC Davis. I'm not there, but that's our local radio station, our campus radio station. So it was a background that I thought was busy enough that, it kind of makes it interesting. And so that's where it is. It's from the KDVS studios. It's their record stacks. I wish I had that many records or had a room in my house that I could devote to something like that, but unfortunately, no.
All right. Well,
Do we want to ask a general discussion questions if anybody has things they would like to suggest for future programs, for research? If they don't feel like doing that today or in the chat, you can obviously email any of us on this call, or Karen or myself or David Block.
Or Caroline.
Oh, Caroline, that's true. Before I forget about Caroline.
She just started.
So basically just to, you know, any ideas for programs, regions you think we should visit, or when we come back to Lodi, obviously, as we will be back, other topics that would be of interest to you, research that we should do that would be of interest to Lodi specifically. This is your time to be selfish and only focus on your own backyard. Correct?
Yes, that is right. Yep.
I saw Dave just shared our emails in chat.
All right. Well, if that's, if there are no questions that are coming up here to the public, again, if anyone has any questions that they want to ask to any of the speakers, if you go to ucdavis.edu, you can find any of us on the UC Davis website. So if you want to ask someone specifically, privately, whatever, you can do that. I'd like to thank all the speakers, and thank you all very much for tuning in today. So,
Thank you.
thanks.
Thank you.
Goodbye, everyone.
Bye.
Thank you. Bye.
Have a good day.